Hello, today we're going to look at how bacteria can cause food poisoning. Now, in our previous lesson, we looked at the growth or reproduction of bacteria. We know that this is called binary fission. It takes place asexually and by a type of cell division called mitosis. Now, the very last image that we drew or looked at in our previous lesson was we had many bacteria dividing again, every 20 minutes while favourable conditions existed. And even though in our previous lesson we had rod-shaped bacteria for the growth or reproduction of bacteria, in this image here we have circular or cockeye bacteria, but it's exactly the same. We start out with an original parent cell, which is the single coccus. Within 20 minutes, that coccus divides by two, half the size of the original parent cell. Within 20 minutes, these two new cells are the size of the parent cell. Cell division occurs again. 20 minutes later, it occurs again and again, and this will keep continuing as long as favourable conditions exist. So during that rapid cell division, we said toxins are produced. Now, these toxins can affect humans and the food that we eat. For example, the toxins can be produced in, for example, raw meat, raw fish, and then we ingest the raw food, or we ingest undercooked food. So when I say we ingest raw food, maybe we're having carpaccio of tuna or carpaccio of beef or steak tartare, or maybe you're eating a homemade mayonnaise with raw eggs, and you ingest the bacteria into the body, and the cell division can take place inside the body once every 20 minutes, and then these toxins are produced and they cause the symptoms of food poisoning. Now, sometimes the food poisoning symptoms can be mild, other times they can be quite severe, depending on one, the bacteria responsible for the food poisoning, but also depending on the health of the individual. People who have underlying health conditions, the food poisoning symptoms can have a more detrimental effect on them. But then there are bacteria, particularly one type of bacteria that we are going to mention as our third example of bacteria in our course, Clostridium botulinum, and you can actually die. One pinhead of the toxin of Clostridium botulinum can actually kill. And it's a very unusual type of food poisoning in that normally when food has been spoiled by bacteria, there might be a bad odour, it mightn't look very nice, but definitely the smell and the taste. The problem with the toxins of Clostridium botulinum, the food doesn't smell any different, taste any different, look any different, so people eat the food as normal. The symptoms then are quite unusual. The toxins attack the central nervous system. Speech becomes slurred, vision becomes blurred, and what eventually happens is there is paralysis of muscles in the body, which can be detrimental to the person's life if the paralysis of the muscles takes place along the breathing mechanism. And actually, what a lot of people don't know, and they're quite surprised, Botox, the injection that people take to line out the wrinkles or get the wrinkles to disappear, Botox is actually a very diluted version of the toxin from Clostridium botulinum. And what happens is you have temporary paralysis of the muscles around the area of the face where it's been put. And personally speaking, I will grow old disgracefully with all the wrinkles in the world rather than to inject a potentially fatal toxin. And that's why I'm into the body. So that is why people need to make sure they go to a very reputable place to get these Botox injections, because you are dealing with something that potentially is very dangerous. If administered incorrectly or the wrong dosage. But students are always quite shocked when they hear, and that's where the name comes from, Botox botulinum. Right, so moving on there. So toxins are these poisonous waste products manufactured during rapid cell division. They can cause damage to the cells of the infected host, like humans and food. And in humans, the toxins can cause food poisoning and sometimes even death. For example, Clostridium botulinum. In fact, the World Cup in 2023 in Paris, there was an outbreak of botulism. Botulism is the actual name of the food poisoning caused by Clostridium botulinum. And one lady lost her life. She'd married to an Irishman actually, and it was in the Chin Chin restaurant in Paris, and they had eaten sardines which had been done in a brine in jars. So obviously something went very wrong, and the poor lady ended up ingesting the toxin of Clostridium botulinum and couldn't get any antidote in time, and sadly lost her life. So again, like I said, there would have been no indication that the fish was contaminated, going by the taste of it, which is why people would eat the normal amount of the food. Now, the thing about these toxins, which we'll go over here, there are two types of food poisoning. So food poisoning, excuse me, is of two types. We have what's called infectious food poisoning, and we have what is called toxic food poisoning. So infectious and toxic, and I'm just going to simplify them here for you. Now again, I have weird ways of learning things. So infectious food poisoning, the bacteria themselves are ingested into the body. How could that happen? Say, for example, you were in a restaurant, you ordered a chicken dish, and the chicken was undercooked. So, for example, you ingest undercooked chicken, which, if it's not cooked in the center, which contains live bacteria. Now, a lot of people think that salmonella is the main bacteria in chicken, raw chicken. It's not. According to the FSAI, the Food Safety Authority of Ireland, you are twice more likely to find Campylobacter bacteria in chicken than salmonella. But I suppose salmonella is the one that we talk about most, so we'll stick with the one that you're familiar with. So you ingest undercooked chicken, which contains the live salmonella. And then the salmonella happen to be mesophiles or mesophiles. And you might remember that their optimum temperature is between 20 and 45 degrees centigrade, and that includes body temperature, right in the middle of that temperature range. So now we have live bacteria ingested into the body, and what happens then is, because they are mesophiles or mesophiles, the 37 degrees centigrade within our body is perfect. And what will happen is they will start to multiply by binary fission inside the body. So we note that reproduction takes place inside the body. However, we now know that as this reproduction is taking place, one cell divides again, toxins are produced. So toxins are produced inside the body. And when toxins are produced inside the body, we call these endotoxins. And have you ever noticed that it takes a few hours after eating the undercooked chicken before the symptoms appear? It could take between 12 and 24 hours. And that's because these bacteria need time inside your body to divide again, and they must generate or produce enough toxins to bring on the symptoms of salmonella food poisoning, like the vomiting, the diarrhea, the high temperature, etc. So two key things we want to note here, and that is, first off, the bacteria are ingested into the body, and then the toxins are therefore made inside the body, and that's infectious food poisoning. And a very good example of infectious food poisoning would be salmonella. Now, the other type of food poisoning is called toxic food poisoning, and this time you ingest the toxin. So, opposite of what happens here, with toxic food poisoning, bacteria multiply or reproduce by binary fission outside the body. For example, Clostridium botulinum reproducing, which we'll talk about when we do preservation, reproduces in tinned food, and that's an anaerobic environment. So the bacteria, Clostridium botulinum, if the can has not been heated to a high enough temperature to destroy all of the Clostridium botulinum, the bacteria multiply inside the tin, and after a while they produce a number of toxins. These toxins will then eventually kill the bacteria themselves, but what will be left behind when you open the can of salmon are the toxins. Now, can I just say that's not going to happen because they're so strict with regards to the canning process. Many years back, I'm going back over 30 odd years ago, there was an outbreak of botulism in an old folks home in England, but that's 30 odd years ago. There are very, very strict conditions now. But if the bacteria multiply outside the body, well, then the toxins are produced outside of the body. And when the toxins are produced outside of the body, we call these exotoxins. You open the tin of contaminated food and you ingest the actual toxin. So just to highlight here again, the toxins are produced outside of the body, exotoxins, and you ingest the toxin. And an example there would be Clostridium botulinum. Now, if we're to name the actual food poisoning, the food poisoning would be botulism. So example, Clostridium botulinum, and it's called botulism. And then salmonellosis. And that's the key difference. Now, use your logic here. Which of the two will the symptoms come on faster? Obviously, with toxic food poisoning, if you ingest the toxin, it goes straight into the bloodstream. In the case of the toxins from Clostridium botulinum, the toxins attack the central nervous system. So the symptoms come on mainly very quickly with toxic food poisoning, whereas with infectious food poisoning, the toxins have to be generated or created or produced inside the body, so it'll take a number of hours. Now, there are exceptions to both rules, of course, but generally speaking, the symptoms take longer to come about with infectious food poisoning. So just to look at our notes here, sometimes toxins are produced inside the body, like salmonella bacteria ingested into the body on, say, undercooked chicken. They happen to be mesophiles, which flourish at body temperature, which is 37 degrees. Remember, mesophiles like temperatures between 20 and 45 degrees centigrade. The bacteria reproduce rapidly inside the body and produce toxins, and that is called infectious food poisoning. In other cases, the bacteria reproduce outside of the body, like Clostridium botulinum, producing toxins inside tinned food in an anaerobic environment, and then the human ingests the toxin, and we end up with what is called toxic food poisoning. Now, I want you to look further down here at our note, and I have a nice summary done of the types of food poisoning, and I'm just going to say to you there to learn those. It's very important, if you're learning about bacteria, that you know the two types of food poisoning. So learn well. So toxic food poisoning, infectious. So the bacteria we'll highlight are ingested into the body, and the toxins are made inside the body, and normal cooking temperature will destroy bacteria that produce infectious food poisoning, and it'll take a number of hours to develop the symptoms, because the bacteria need time to make them. Whereas toxic food poisoning, the bacteria reproduce outside of the body, so we have what are called exotoxins. You need very high temperature, so normal cooking will not destroy the toxins made outside the body, and the symptoms can develop quickly. So Clostridium botulinum can bring about toxic food poisoning called botulism, and Salmonella bacteria can bring about salmonellosis, food poisoning caused by salmonella. So just to make sure that you know the two types, and what you have in that table there will cover you well. Right, going back to our handout, or chapter I should say, here. So that's what the toxins can do. They can bring about food poisoning, infectious food poisoning if the toxins are made inside the body, toxic food poisoning if we ingest the actual toxins themselves. Now, this rapid cell division that we've talked about going on every 20 minutes, it can't last forever. Remember, I emphasised that the binary fission or the cell division will occur every 20 minutes as long as favourable conditions exist. But at some point, unfavourable conditions will come about. Now, we don't have to learn this for home economics, but I always bring it up because biology students do it, and it's also good for the home economics student to understand the different stages in the growth pattern or reproduction of bacteria. So initially, when bacteria are present in the food, they need time, shall we say, to settle in, and we call this the lag phase. Then suitable conditions or favourable conditions, all five: food, warmth, moisture, correct oxygen level, correct pH. And during this phase here, the bacteria are increasing madly in number. They're dividing again every 20 minutes. However, then comes a stage when conditions start to change from being favourable to unfavourable. Because there are so many million of bacteria present now in the environment, they are starting to run out of food, run out of moisture, run out of oxygen, if that's what they require. And also remember, as they have been dividing by binary fission, lots and lots of toxins are being produced. So these toxins are starting to kill off some of the bacteria. So during the stationary phase, bacteria continue to be new bacteria continue to be created, but whatever number is being created, the same amount is actually being killed by the presence of toxins, by running out of food, moisture, oxygen, etc. Then comes the point when conditions are extremely unfavourable. We call this the decline phase, where conditions are, so we're talking here at this stage, very unfavourable conditions for growth. And with very unfavourable conditions for growth, like a lack of moisture, a lack of food, a lack of oxygen possibly, and also the high amount of toxins. These are all very unfavourable conditions. So what will happen is a lot of the bacteria will start to die. And if you notice, if you've ever had food poisoning, initially the bacteria which you ingested on the undercooked chicken, for example, they take a while to settle in your body, the lag phase. Then they start to divide again, by binary fission, producing lots of toxins. And then the toxins cause or bring about the symptoms of food poisoning. So you feel extremely unwell for maybe a number of hours or even a day or so, but then you're feeling much better. Why? Because the bacteria start to die within your body. They've been so greedy, multiplying madly, producing lots of toxins, that eventually what happens is the toxins that they've made to cause you to become ill or cause the symptoms of food poisoning, eventually those toxins kill the bacteria themselves. However, there are some bacteria, they're called gram positive. And around where I have that green asterisk there, it's at this point when there are very unfavourable conditions for growth. Some gram-positive bacteria, now I'm personalising this, some gram-positive bacteria say, hold on here, things are not looking good. So what they do is, rather than die, they protect themselves by forming what are called endospores. Now, endospores can also be called bacterial spores. So endospores or bacterial spores. If the bacteria are gram positive, and a reminder that gram positive bacteria have no flagellae. So these gram positive bacteria are able to produce the bacterial spores, but they will only produce the endospores or bacterial spores when conditions for growth are unfavourable. So when conditions for growth are unfavourable. And just to remind ourselves again, what do we mean by these unfavourable conditions? Toxins are building up in the environment. Food is running out. Oxygen is possibly running out as well. Moisture and so on. And when the conditions are unfavourable, what happens is spores can form within bacterial cells, but only the spores only bacteria that are gram-positive. But the bacteria must be gram-positive. So you need to know the following diagrams. So I would take a rod-shaped bacteria, no flagellae, and it's a gram-positive bacteria. And inside that bacteria, a spore develops within the walls of the bacterium, which is a singular bacterial cell. And that's why we call it endospore, because it forms within the actual bacteria itself. And what happens then is the spore gets larger. So the spore gets larger, and then protein layer forms around it. And so this is our endospore, grows in size. And this protein layer forms on the outside. And when the spore has fully developed, the walls of the bacterial cell break down, and the spore is now, I'm getting the wrong color there. Oops, there we go. It's a problem to start bringing in color, but otherwise it would look quite boring to look at it. And the spore is released. So when the spore or endospore, bacterial spore, so when the endospore or bacterial spore is fully formed, it's released as the walls of the bacteria break down. So just to look there again. Bacteria divide rapidly when conditions are favourable. In fact, they can divide so quickly it can happen every 20 minutes. Eventually though, the conditions become unfavourable as lots and lots and lots of bacteria are produced. Conditions start to deteriorate. Food is running out, moisture is running out, oxygen is running out, toxins are building up in the environment. So bacteria are starting to die, but it's survival of the fittest. Some bacteria, gram-positive bacteria with no flagellae, they have a high resistance to antibiotics. What they start to do is they start to form spores within the bacteria themselves. As the spore gets larger, a protein layer forms on the outside. Eventually, when the spore is fully formed, the walls of the bacterium break down and the spore itself is released. Now, the thing we should know about these endospores, also called bacterial spores, they're just bacterial spores that form within the bacteria, and that's the word bacteria. We can say that they are tough, they are dormant in that they are asleep, and they are extremely difficult to destroy. In fact, in canning of fish and other protein foods, 121 degrees centigrade for 15 minutes is the temperature the can is subjected to. Do you know that's equivalent to 5 hours of boiling? Now, there is nothing I know of that would survive 5 hours of boiling at 100 degrees centigrade, and that is the equivalent of 5 hours of boiling. And that's the kind of heat or temperature that is needed to kill, say, the spores of Clostridium botulinum. So what can happen then is, if those spores were not destroyed, and say, for example, canned salmon was not heated to 121 degrees centigrade for 15 minutes, and some spores survived. Imagine our tin, completely sealed, anaerobic, lots of lovely, moist, protein-rich fish, perfect pH, perfect temperature. And what happens is, these dormant endospores, as soon as they stay asleep, they stay dormant until such a time, if things pick up, if their environment suddenly becomes conducive to growth, they start to multiply again. So can you just imagine, inside in a tin of tuna, for example, where 121 degrees centigrade for 15 minutes has not been adhered to. And what will happen is, these spores of Clostridium botulinum, they start to divide again, and you end up with lots and lots of new bacteria, and they divide again, and they produce toxins inside the tin. After a while, there'll be so many toxins in the environment, all the bacteria will end up dying. But note, even though all the bacteria are dead, what's left behind in the can? The actual toxins. And what happens then is, you open the tin, doesn't smell any different, doesn't look any different, doesn't taste any different. And like I said earlier, one pinhead of the toxin of Clostridium botulinum is enough to kill. Fascinating stuff. It's very, very interesting. So there's our log phase, stationary phase, decline phase. It's just good to know that, and to know that during the decline phase of the growth pattern of bacteria, this is when endospores of gram-positive bacteria are formed. Right, moving on there to our chapter, formation of an endospore. So bacteria can't multiply rapidly for long as they start to run out of food, overcrowding, competing for air, food and moisture, toxins building up in the area. And so what happens is, the gram-positive bacteria produce these endospores, or bacterial spores within the cell. But it's important to note that the conditions have to be unfavourable for the endospores to be formed. No food, no oxygen, no moisture, and the presence of a large amount of toxins. So NB, you need to learn the formation of an endospore, and that means include diagrams. So an endospore begins as a small area within a bacterium, which is a single bacterial cell. The endospore increases in size, and a tough wall of protein forms around it. Eventually the wall disintegrates and the spore is released. And that spore will, and so we'll just say here, the spores or endospores will remain dormant, as in inactive, until such a time favourable conditions exist again. So just to summarise there, make sure that you are familiar with the two different types of food poisoning and how they're caused: infectious and toxic food poisoning, and also to be aware that bacteria that are I suppose, gram-positive with the no flagellae, they have the ability to produce these spores to continue on their existence and survival of the fittest. So that completes today's lesson.